following is a conversation with Dr. Laura Craven, lead research and development scientist at EnteroBiotix, a biotechnology company developing novel, full-spectrum therapeutics for the benefit of patients around the world. Dr. Craven also holds a PhD in microbiology. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. Excellent. So, how did you get into the field of the microbiome?
It was, I think, a happy coincidence that I got into it. So I had finished my undergraduate degree in microbiology and was thinking about doing a master's degree.
So you've loved bugs for a long time.
I have. I've always really been interested in bacteria, but I didn't really know what I'd want to do for a master's project, but I knew I was interested in the probiotic field. So I approached a professor at the university to say, hey, do you have any projects available? And they said, oh, I'm so sorry. We've actually just offered a position to someone. If they say no, we'll get in contact with you.
Okay.
The next day, I get an email to say, they've turned it down.
No way.
Do you want to come in for an interview? And
Fate.
I was like, exactly, I know.
Fate.
So I came in, talked about the project. I thought it was going to be probiotics, and it was an FMT project. And I was like, oh, I've never heard about this.
You hadn't heard of it once?
No.
Oh, interesting. Mm-hmm. When was this? Just out of interest?
This would have been summer of 2015.
Yeah, so still new.
It was.
FMT was new on the scene.
Yeah.
Okay.
So I had never heard about that. They were telling me what this project was going to be, and I said, I really want to be a part of this.
I really want to be a part of understanding how stool samples can treat disease.
Exactly.
Yeah. I felt exactly the same. As weird as it may sound, when I read that paper, the New England Journal of Medicine paper, Duodenal infusion of donor feces for recurrent C. difficile, and I saw the graph, and post-production will pull it up, that compares the 3 different treatment arms. And I saw that those who had the FMT were staying out of hospital 90% of the time at 8 weeks versus 20-30%. I just thought, this is going to be huge. Mm-hmm. This is going to be massive. And I was also intrigued because it was like, sorry, we're doing what? Moving stool from one person into another? So it'd be interesting to hear what you thought when you heard of it.
I had never imagined anything like that before. I think in the field of probiotics, I thought it's, single strain, grown up culture, purified some way, made into some kind of formulation given to someone. I had never thought about an approach of taking healthy stool and giving that to somebody else.
Yeah. So what happened next?
I did really well in the interview because they offered me the position. And I started a couple months later. So it was a kind of a joint project between the Department of Microbiology and the Department of Infectious Disease. And I got to have a really great experience with lab work and clinical work to see the impact that FMT could have. So
walk me through from start to finish succinctly. Because I know PhDs are
it's a long time.
A long slog. And it's kind of funny because you interview someone at the start of the PhD, or you speak to them at the start of the PhD, and they're buzzing. They're like, this is amazing. I'm going to contribute. And then by the end, they're like, don't ask me.
It's a big commitment.
Don't ask me about the thesis. Don't ask me about the defense.
I think never ask when you think you're going to be done. That was the question. I didn't like at the end. Don't ask me when it's going to be done. It'll be finished when it's finished.
Yeah. It's the same in business though, isn't it? You got a milestone and everyone asks you all the time. Mm-hmm. And it's like, I will let you know as soon as
it's not something I'm keeping a secret. When it's ready, I'm going to tell everybody.
I think about this. It's funny because, the investors want the company to be successful, but, I spend all day, every day, literally doing this and thinking about this.
Yeah.
And for some of these milestones, like as soon as I know, I will let them know. I promise you, I will, as soon as I hear, I'll call the ones that are up. But they still ask every time. Like, you done it yet? And it's like, no. I'll let you know. Oh yeah, okay, fair enough. But they're just excited. So tell me about the PhD then. And maybe before that, what was it about the microbiology undergrad that was like exciting you, and made you want to do academia rather than, find a job in industry or something like that?
I think about just how closely bacteria interact with humans was a really exciting part of it. A lot of my undergrad was really focused on pathogenesis, and we had a few lectures that were on probiotics and the beneficial effects of bacteria. And that's what really drew me in, is that actually these can be doing a lot of work for us and they can be help, not a hindrance.
Yeah.
I wanted to explore that.
So it's about them being beneficial rather than harmful,
Yes.
which historically has gone against everything.
Anything I'd ever
taught.
learned up till that. Yeah.
Yeah. Their pathogens are bad. Clean your hands.
Yep.
Cool. So walk me through the PhD then.
So it had kind of two halves to it. So there was the clinical component where there was 2 FMT clinical trials that I was a part of. So one of those was in non-alcoholic fatty liver disease, and that was the first study that we did. And the second was in multiple sclerosis.
So should we just take a second to discuss FMT versus IMT?
Yeah.
So for the listeners who've listened to Biome Bites, episode number 1, I think, hopefully that's 1, they will have learned about IMT. But I think it was you who coined the phrase. Was it not? It was you.
It was
Or
us.
was it me? Was it us? Was it you?
I was going to credit you for...
Oh, you were credit me?
Yeah.
Thanks. I appreciate that. I was going to credit you. Well, that's good
very
teamwork.
kind.
That's good teamwork.
Yeah, I'm good. It's us.
It's us. So we published it.
Yes.
Yeah, and we may have been the first. Although there was the University of Minnesota crew who also were calling it something different. Just microbiota, I think, rather than intestinal. So walk the listener through, how it was historically called one thing and how we're trying to call it another thing, but we still by default call it FMT. It's really annoying. IMT.
It gets stuck in my brain. But FMT would be fecal microbiota transplant, and that's what it's been called, I don't know, since 2011, whenever that...
Maybe before.
Yeah, before.
Originally, actually, I think it was called fecal bacterial therapy.
Okay.
From the Ben Eisman case report in 1958, who had those 4 patients who weren't getting better, and he was just like, yep, I'll decide. I'm the chief surgeon. I'm going to do this procedure, and they got better. 1958 journal surgery, fecal bacterial therapy, I think it's called. Yeah.
That's an interesting history.
Yeah.
But, it's got, with the word fecal associated with some negative connotations.
Yeah.
And I think that's been a really big barrier in healthcare for patients and for doctors to bring it up because there's that ick factor associated with fecal. And as time progresses and things get more refined, it really isn't reflective of fecal anymore, which is why we proposed intestinal microbiota transfer.
Yep. And that's what we try to use now.
Yes,
IMT. Because I mean, we can talk about our product. I'm jumping ahead now because you're now working at a company called EnteroBiotix, and I am too.
Disclosure. Yes.
We'll, our product. Sure, the starting material is rigorously screened stool samples from really healthy people, but it would be impossible to tell unless you knew, because it's this off-white, odorless, free-flowing powder.
Yes, and it looks nothing like that starting material anymore. It's been processed, refined, formulated. It's not the same.
Yeah. So I think, but in another respect, if you don't have the kind of technology and capabilities and know-how and intellectual property that we have at EnteroBiotix, you're probably not likely to be able to create a product like that. So maybe people still feel they need to call it FMT because it's this brown liquid,
I'll probably think of it as FMT as I discuss my PhD, because I think that was much earlier on than we are now.
That makes sense.
But IMT is more what I think of when I talk about EnteroBiotix.
So this sounds like very pioneering work, because in 2013 we had one study which was in C. diff. But in 2015 you were talking about multiple sclerosis, so an autoimmune disease that affects...
That came a little bit later,
right?
But it was NAFLD that came first.
NAFLD. So let's talk about NAFLD then. So that's a liver problem?
Yes, non-alcoholic fatty liver disease. So there's an accumulation of fat in the liver that can be a metabolic related disorder or an obesity related disorder. And over time, that can progress to liver cirrhosis. So it's really key at that stage to try and reverse the accumulation of fat. And it is a stage of the disease that is reversible. So we were looking to see if we could reduce the percentage of fat in the liver in these patients by doing an FMT from a thin and healthy donor.
And what was known at the time about the microbiome's involvement in NAFLD? What was the hypothesis?
Yeah, is that the Turnbow paper? The obese mice.
Oh,
and you could
the
take
classic.
yes. So I think that was one of the big things.
mouse.
Yes, that was a big thing that was known then. But we already knew that the liver is the first organ to encounter the blood that's gone past the gut through the portal vein. So there is a really big connection between the gut and the liver, and that was well known before that study took place.
Got it. So somebody within, probably the professor, was it a supervisor who came up with this idea? He was like...
It was a big team. Like, it's a collection of people who came up with this.
And I suppose, just to kind of put myself in their shoes, they probably thought diet reverses or stops the progression. So maybe instead of having the patient change their diet, we can just give them microbes from someone who's got a good diet?
Yeah.
Essentially?
Pretty much.
Yeah.
And is there something that we could transfer that would be beneficial? So there's plenty of people that don't have non-alcoholic fatty liver disease, but may be similar to a population of patients that does. So what's the difference between people who don't develop that and people who do? And maybe that's the microbiome.
Got it. Yeah, right. Okay, got it. So they've got the same lifestyle, similar genetics. One gets it, one doesn't.
Yeah.
Is it the bugs? Mm-hmm. Very interesting. So describe the trial design then. What was the thinking?
So we're going to talk about the meaning of placebo and FMT or not. So this was an autologous.
Yeah, this is a good one.
Autologous. Yes,
okay.
This was an allogeneic versus autologous study. And I think, knowing what we know now, I don't know if autologous would have been chosen as the control group. Yes. So an autologous FMT, which I don't know is as popular now, would be receiving your own stool back. Yep. So this would be patients donating a stool. It gets turned into an FMT and given back. And it was a randomized, double-blind controlled study. So they didn't know which treatment arm they were in.
Interesting. So I guess it's blinded, but it's not a true placebo insofar as it's having some sort of physiological effect.
Yeah, and I think there probably is some kind of effect from receiving any FMT, regardless of the source. So,
just for listener, why might that be?
There's definitely an immune response, I think, that happens when receiving that kind of heavy dose of bacteria. It's probably delivered in a region of the gut that it didn't originate from. So this would have been delivered into the small intestine. This is material that came from the colon and was passed as feces. So it's not the same composition of bacteria that are normally there. And that can definitely elicit some kind of immune response.
Got it. So for the listener, they decided that autologous was a placebo, but classically the placebo is like an inert.
It really should be.
Should be.
And at the time, I think that's what people genuinely believed. I think it had the best of intentions that this was a proper control group.
Yeah, nothing would happen, right. Oh, Okay. And I suppose the patients, of course they didn't know.
No.
There's no sixth sense being like, oh my God, that's mine. Or, oh my gosh, sorry, that.
No.
Oh my gosh, sorry.
And it was delivered via endoscopy, so they were sedated when they received it too.
Okay.
So that I don't think they'd be able to look at it and have a guess that was theirs.
Yeah. And when you say endoscopy, was it going through the nose and into the intestine, or was it going up the back passage?
No, it was fed through the mouth and then into the small intestine, so it wasn't through the nose.
Got it. I understand.
Yeah.
Okay, so it was an endoscopy delivered straight to the intestine.
Yes.
Interesting. And when you say they were sedated, they weren't asleep. They were just like sedated, as in they were given a lot of sedating medication and they weren't really sure like what was going on. Okay.
Yeah, put them in a more relaxed state to be able to have that kind of procedure done safely.
And perhaps a big question for you is, what was done about the diet in these patients? Like, did you give them dietary advice before or after?
There was supposed to be no dietary advice. So we really wanted to see what would happen if they changed nothing about their lifestyle. So we did collect data about their diet and their exercise to try and see if someone had changed and if that had any impact on the data, but they weren't supposed to change anything.
Because a lot of the thinking now around cravings and food preferences and choices and that kind of thing, I believe links to, the microbes have changed and they're influencing your cravings somehow.
In exercise too, I was reading that paper that the composition of your microbiome makes you more or less likely to want to exercise. I think that's really interesting.
It's not me, it's my microbes. Sorry, I can't help that. I wasn't that sure about that one, to be honest. But it could be true. I mean, we've done a little office experiment where we give everyone unlimited green juice and fruit and stuff like that. And I think originally everyone was like, no, I don't want that. That's terrible. Why is James doing this? But then a lot of people now drink it quite frequently, and some people say when they don't have it, they don't feel good. So my thinking is that maybe the microbes have adapted to that daily green fruit stimulus. But anyways, so what did you see in the study?
So we didn't see a change in the amount of fat in the liver of these patients. And that was the primary outcome of that study. The really interesting thing that we saw had to do with their intestinal permeability. And we saw that intestinal permeability
Your eyes lit up
improved. I'm so excited about it because that's my favorite thing from the study. And I love talking about it. But that improved following FMT in the allogenic group.
Allogenic mean from the
lean, healthy donor.
Lean, healthy donor. Was it a single donor?
I'd have to double check. There might have been more than, it wasn't a pooled product, but there might have been more than one donor in that study.
Understood.
Yeah.
And how was the donor selected or the donors selected?
You can read Craven et al. 2017, the extended screening criteria that we used for that study. It's published separately, but there is a protocol for how we screened. But it was a mixture of biosafety screening questions, lifestyle questions, but a heavy emphasis on metabolic disease and ensuring that we had a donor that didn't have risk factors for metabolic disease.
Did you measure any were there clinical assessments of the donor's metabolic status, or was it mainly just questionnaire based? Like, did you look at liver function?
Yeah, those would have been measurements included, A1C, things like that too. Yeah, so we did look at that in the donors to select for people that should have been resilient to metabolic syndrome, essentially.
Great. So, your favorite topic, leaky gut.
Yes.
Intestinal permeability. Go wild. Tell us everything.
Yeah. So,
what is it and why is it important?
So the cells that line your intestine should form a barrier so that there's regulated transport of material from within the intestine into your body. When you have leaky gut or increased intestinal permeability, there are metabolites and material that can pass in between the cells that make up that barrier through paracellular transport. It's not regulated. And a lot of those materials can be highly inflammatory to the body.
Right.
So ideally, you have a gut barrier that is intact and functioning, as intended. And in addition to having those cells that are tightly bound together, there's also the mucin layer, and there's quite a few factors that go into the intestinal barrier.
So the intestinal barrier is an anatomical, physical thing.
Yeah.
The cells are not stuck together, but
they are stuck together. So there's things called tight junctions which hold cells together.
Right.
And if you have less tight junctions, the less well held together these cells are, the more things can pass unregulated through the intestinal epithelium.
Describe to the listener what the things might be that can pass through that we don't want.
Things like LPS, lipopolysaccharide, that's something that's made on the bacterial cell membranes of gram-negatives, is a big thing like that. So LPS is a very potent inflammatory molecule. And it's not something you want in high quantities floating around your body. It's quite bad. Yeah. And when that is able to pass through, it's an inflammatory state, essentially.
So E. coli must have LPS, right?
Yes.
Massive side tangent. Mm-hmm. Do you know someone called Wim Hof?
Yes. Yes, the breathing method.
Yes. But do you know that Wim Hof was injected with bacterial endotoxin?
I did not know.
Is that synonymous with LPS, endotoxin, LPS?
Yes.
Same thing, right?
Yes.
And nothing happened.
How much was it?
A lot.
Wow.
Enough to make you sick.
Mm-hmm.
So what happened originally, I think, was they were analysing him for everything. They withdrew some blood and ex vivo, so outside the body, put some endotoxin in to see what happened. And there was a big antibody response.
As there should be.
Yes. Yep. And there must have been some other things as well that were quite unusual. I think it just neutralized it. I think it did something to it. I don't know the exact ins and outs.
Yeah.
And then they gave it to Wim Hof when he was doing his thing, and nothing happened. No systemic inflammatory response, nothing. And then they were like, "Well, you're the Iceman. You're special." Yeah. He was like, "No, I'm not. It's just my method." So then they got two groups of people. One had the Wim Hof method, one didn't. And they both got the endotoxin, I think, and the people that did the Wim Hof method didn't get sick.
I don't know how they're getting ethical approval for this.
I don't know. I mean, this is honestly what I read. I mean, a lot of people laugh at Wim Hof. I was one of those people, but now I'm such a believer. I'm total Wim Hof fanboy because a lot of the work that they've done on Wim Hof has been published in really high impact journals. So, sorry, I've taken us so off piste. I'm really sorry. Let's go back to...
Maybe he has amazing...
Do you do Wim Hof method?
I do not. I've never tried.
Are you skeptic?
I don't think I know enough about it to even attempt.
Oh, God. It's well, actually, we're having someone come into our office to do a breathing workshop.
I saw that. Yeah. Is it the Wim Hof method?
Yeah, I think...
Is it?
I think... I don't know if it's the Wim Hof method, but I think it's a variation of Wim Hof to manage stress. And Wim Hof claims you can tap into your autonomic nervous system and your endocrine system and your immune system. Yeah. So this person's coming in.
I won't make my mind up until I've tried it.
Yeah.
Yeah.
Well, good, because it's amazing. it really is. It's incredible. So back to your favorite topic. Sorry.
Intestinal permeability.
Yes. So why do the tight junctions and the intestinal permeability change? What's going on? Because it doesn't sound like it's a good thing.
It costs a lot of energy for those cells to maintain tight junctions. So it needs a source of energy to go towards keeping them up. But one of the sources of energy that the intestinal epithelial cells use are short-chain fatty acids. A big one of those is butyrate. So if you have altered compositions of short-chain fatty acids, those cells may not be able to produce tight junctions as well, and that can cause the leaky gut.
What does the listener have to do to maintain a strong gut rather than a leaky gut?
Yeah.
Does that make sense?
Yeah, no, that does. A lot of short-chain fatty acids are derived from dietary fiber. So
Nice.
One great thing to do would to be eating fruits and vegetables.
Yep.
Yes.
Put your fiber intake
Yes.
up. Got it. So how many problems are associated with leaky gut that we know of? And can I just add one more thing? Is before we spoke today, I went back and I typed in leaky gut and I had a look at stuff. And I found some articles from like 15 years ago where they were just like, leaky gut doesn't exist. It's not a thing. It's not a problem. It's just a myth. It's like because I think back then it was maybe more of one of these topics that was more naturopathic medicine than, does that make sense?
Yeah.
Than sort of modern medicine.
Quantifiable, measurable,
Yeah. So, how do we know if someone's got leaky gut and what have we associated leaky gut with, if that makes sense?
So how do you know if someone has leaky gut? You can measure. So one of the most common tests, the gold standard would be doing a lactulose and mannitol test. You drink a solution of these two sugars and you measure the permeability of your small intestine essentially by looking at the amounts that are excreted in urine. And you can use the ratio of the two to predict your gut barrier function.
Right. And the thinking there is that they're not supposed to pass through, or some of them are supposed to pass through?
One can and one shouldn't, and you can use the ratio of the two to measure that.
Perfect. And the thinking there is that if it passes through, the one that passes through should be excreted.
Yes.
Because it goes into your blood and then your kidneys filter it out and you pee it out. But the one that shouldn't.
Yeah.
It should come out your back passage.
Exactly.
Right.
It should remain in the gut and be excreted in stool versus being taken up by the body and excreted in urine.
Got it. But if you see, and it must be the lactulose.
Yes, because that's the larger molecule of the two.
If you see the lactulose in the urine, that's a problem.
Yes.
Right. But that sounds quite complicated for, average Joe or Julianne in the street.
Yeah, that would be a test that requires expertise to analyze. Absolutely. That I don't know how widely available that would be to measure yourself.
Got it. So a lot of people that say, I've got leaky gut, they've probably not had it assessed. They just feel like they do because of the symptoms they've got, something like that.
Potentially.
Yeah. So how does FMT, IMT have an impact on leaky gut? Like what is the thinking there?
So you're changing the composition of bacteria and other microorganisms that are there. And one of the theories is that you're changing it to a composition that's better able to make short-chain fatty acids so it can feed those intestinal epithelial cells to maintain the strong barrier between them.
So why not just give short-chain fatty acids in a drink?
I think people have tried and it just hasn't had the same impact.
Why does it not work?
Yeah.
Because in ulcerative colitis, so butyrate is important for the, well, you talked about tight junctions, but it's just important for gut health in general, isn't it?
It is. It's a source of energy, and it can be used for a whole wide variety of things.
Yeah. So the thinking in ulcerative colitis, which is this form of inflammatory bowel disease that exclusively impacts the large colon, was there's a reduction in butyrate producing bacteria, and as a result of that, the mucin starts getting degraded because that's a source of energy for some of these mucin degraders, and then as a result, there's this shift towards pro-inflammation versus anti-inflammation or whatever. So somebody said, well, I'll give these patients butyrate enemas. So they filled this enema up with butyrate and they scooshed it in, and it didn't work. So is there something else going on, or is it because it's just like a transient thing and you need the bugs in there?
I think it helps to have the bacteria there to continually to produce that versus just a one-off treatment.
Yeah. So how do you think about bacteria then? Are they like these factories that just, pump out the good stuff if you feed them the right stuff?
Yeah, I think that's part of it. And it's amazing. There's, different chains of bacteria where one's going to make one metabolite that the next one can use and the next one can use. So there's lots of symbiosis that takes place in the gut as well, which is really interesting.
So what you're saying is some bacteria exist almost to serve another bacteria's needs.
Yeah.
And then that impacts on another bacteria. Yeah.
Like you definitely need a group altogether to have some of the intended effects that we're looking for.
Oh, crazy. So bringing us back now to your study, you saw something relating to intestinal permeability that was really exciting. So what did you see?
So we saw intestinal permeability decrease, which means the gut barrier function improved. There was less of this transport of unintended molecules across the membrane.
Wow. So let's zoom in then to the gut in patients with NAFLD. Why might that be important for patients with NAFLD?
You're trying to reduce the amount of inflammation in the liver, and hopefully you're going to reduce the amount of fat accumulation that's occurred there. So one of those things can be just the inflammation from having leaky gut.
Yeah.
And there's a few other hypotheses about what the gut microbiome is doing that's feeding into that. One could be having a large proportion of gram-negative bacteria. So there's just more LPS floating about. And if you also have leaky gut together, that can cause that fat accumulation. Another is endogenous alcohol production. So you might have a composition of bacteria that are able to produce alcohol, and that can be, pass through the body and cause the inflammation in the liver as well. So it's not alcoholic fatty liver disease, but there still could be alcohol playing an impact.
Let's talk about endogenous alcohol production. It's not something I know much about at all. So what is it you're saying? You're saying that there's some bacteria that produce alcohol?
Yes. Through their metabolism, there are some, and that could also, other microorganisms too, that can include yeast and things like that. Okay. But they can take, a glucose source and convert that into alcohol. And it could be in minuscule amounts, and that's enough to cause this inflammation. I think there's some case studies that document a very large amount, which I don't know how likely that is to happen. But there are people who have, said, I drank, a glass of juice and then I was drunk soon after, and it could be from an overgrowth of yeast.
You've just reminded me of that. It's called Brewer's syndrome or something like that, isn't it?
Auto-brewery syndrome, maybe?
Auto-brewery syndrome. They're just drunk all the time. That's mad, isn't it?
I can't imagine having to live life like that. It'd be really hard.
I very rarely drink alcohol. That would really be hard for me.
Yeah.
Can they drive? No, they probably can't drive.
No, I don't think so.
They're not allowed to drive.
I'm sure they have to measure their blood alcohol or something to make sure it would be safe to drive. I don't know how many people that impacts, but I think there's still very small amounts of alcohol that can be made by the microbiome that by no means has that kind of impact as auto-brewery syndrome.
Fascinating.
But it could be something like that. And I think there's all sorts of factors that can play into having non-alcoholic fatty liver disease. I don't know if everyone has the same set of factors, but that could be part of it.
That's really interesting. I mean, has anyone tried to create a probiotic that gets you drunk? I don't know why you would do that.
I don't.
Why you do it, I don't know, but someone might have done it.
Part of the definition of probiotic is to have,
health
benefits.
It is, actually, that is the definition, you're right.
So I don't know if that would quite fall under that category.
Okay, so we'll talk about probiotics in a minute, but I think it would be really interesting for the listeners for you to describe the process of getting those results and the excitement that the team had finding something interesting. So because it's blinded to
It is blinded.
Yeah, which means that you don't know. So how did you uncover the result? And how did the excitement permeate through the team?
So we didn't know this result till the very end because all the samples were analyzed as one batch. So it was, I think, nearly 2 years after the study started, we got this data back. So in real time, we were getting data back on, the bloods, the MRIs that were looking at the percentage of fat.
Right.
So we'd known some of that data relatively quickly. What we didn't know was this piece until pretty close to the end.
And the clinical data wasn't setting anyone's world on fire.
No, it was still really good data to collect. It's a pilot study. It was looking at safety, and it showed it was safe. So that data is still really valuable.
Yes.
But in terms of symptoms of the disease, it didn't really seem to show any difference after the FMT, with the exception of intestinal permeability.
Now, again, just before we jump into that result, what were the patients? Was there high patient acceptability? Were patients put off by the term FMT?
I think there definitely were.
Some put off.
Yeah. So I think there was about just over 20 people in that study, and it still took 1.5, 2 years to do all that recruitment. So it takes a long time to convince people, and there wasn't the same wealth of knowledge about FMT, IMT in 2015 as there is now. So I think it was a bit more difficult to convince people then than it probably would be now.
That's really interesting. I mean, you've been in the field now since 2015. How have things changed from like a public engagement, societal understanding point of view?
Almost everyone I know has heard of FMT or IMT, and it wasn't from me, which I think is really interesting. But someone's heard it on the radio, on a podcast, read a news article. A friend of theirs has talked to them about it. So whenever I tell people I study IMT, they're like, oh, I've heard about that before.
Nice.
Can I ask you some questions? Like, absolutely. So I don't have to sell it anymore like I used to. Because there was a time where I'd tell people this is what I did, and they're like, oh, that's really strange. I don't know why you'd want to do that. And now everyone's so excited about it.
Yeah, they are.
And then the gut microbiome, they're like, wow, that's really interesting.
Yeah. Well, when I got started, people literally laughed in my face. I'll never forget it. I went to this business networking event in Aberdeen, and we were doing the sort of, hi, how are you doing? My name's Linda. What do you do? And there were these two men who worked in the oil and gas industry, and I explained what I did, and I promise you, they both burst out laughing in my face. And they were like, you want to do what? And they just burst out laughing, and I was like, wow. That feels really bad. No. People laughing in my face, but
I was going through customs in America to go to a conference, and the customs agent was asking me why I was there, and I told them, oh, I study fecal transplant. I'm here to, attend this conference and learn more. And he's like, oh, I don't know why you'd want to do that. Like, and I was trying to tell him why it was interesting, but also really trying to get through customs without upsetting this man. Yeah,
because it can be very...
And at the end, he just looked at me. He's like, I really hope you find your dream one day. like what I was doing now just wasn't it.
It's quite rare for them to engage in any...
I never have had that much
dialogue.
interest. Yeah, he was really... And it was like midnight, I think, flying into Boston, but he just really wanted to chat about it. But at the end, he seemed disappointed I hadn't, realized my full potential. And one day I'm going to get there and he's going to be really happy for me.
Oh my goodness, that's really funny. It's changed a lot, hasn't it? I mean, everything's gut health now. You go into the supermarket and there's gut health specific products. Everyone's asking about probiotics. FMT, IMT, people know about it. Yeah. A lot of people want it.
Yeah, I think people ask, it's like, oh, when is this something, that I could buy? I've got,
not yet.
Gut problems, maybe IBS, IBD. This would be really interesting if I could try it. And it's like, well, not there, but that's the dream.
Soon.
Yeah.
We're getting there. We're getting there. So talk me through the excitement and how you were like, wow, we found something. We found something.
It is exciting.
Was it like that? Was like,
yeah. No, absolutely. I think we would have had more excitement if we'd seen other parameters change in the study. Yeah. But it felt like a good win to see that changing the gut microbiome had that effect in the gut. And that was the goal.
So going into the study, you had some hypotheses, presumably.
Yes.
And was one of them that you would improve intestinal permeability?
Yes, it was.
Fantastic. But
it's the first publication that shows that you can improve intestinal permeability with FMT. As far as I'm aware. Yes.
And it's Craven et al.
Yes.
It's Laura Craven et al. Whoa.
It is. And a team of very talented individuals as well.
Absolutely.
Yes.
Shout out to all of those very talented individuals.
Yes.
But okay, so this could be historic. You're going to get a Nobel Prize one day?
No, I don't think so.
I know. What if it turns out that intestinal permeability?
It's not solely my idea. This is a team of people.
Yeah,
it's not just me. Well, I'm listed first. There's a lot of credit to go around.
Yes. So the holy grail, I guess, for research is a hypothesis linking a mechanism with an outcome that ideally improves someone's life.
Yes.
That, to me.
Yeah.
So if the study had gone a step further and linked the permeability to less fat in the liver.
That would have been a mechanism of action. So we didn't get that out of the study.
But do you believe that might happen in a bigger study, longer term follow-up, difference in dose?
Definitely. It was a very small study that was, again, designed for safety purposes, and it passed that.
Yeah.
Larger study, longer follow-up period.
Yeah, and dose.
It takes a long time for NAFLD to develop. It might take a long time for it to improve as well.
This makes sense. So how did you define dose in your study?
Dose at that time, and you'll laugh.
At that time, caveated.
I just think it needs context because we've learned so much since then.
Yeah.
But at that time would have been grams of stool.
Well, there's still people out there doing grams of stool.
There are still people doing that.
Tell us how we moved on. Yeah.
So I think now people would try and recommend, number of cells, CFU per dose versus grams of stool. Grams of stool and the concentration of bacteria can be very variable between different donors or within a donor over time. So there's less standardization, I think, when you do grams of stool. But at the time, that seemed to be very well accepted that you said, oh, what's your dose? It's this number of grams of stool. And people are like, yes, I get that.
Was it 50 grams?
No, it was less than that.
It was less than that?
Yeah, it was
like
less
30
than that.
or something?
I think it was actually quite low. I'd have to double check.
Yeah, that's interesting.
Yeah.
Because there were so many 50 grams just appeared. And everyone was like, 50
it just stayed.
grams. 50 grams is the dose. It's like the 4 to 6 hour window between collecting a stool sample and
the
processing.
leg time.
I think I was there when that came up. I think I was there at I was there. I was in Rome when they were having that discussion that became the consensus guidelines.
This was a fresh stool study too.
Right.
Yeah.
Okay. Oh, interesting.
So this was before the big body of evidence that frozen was acceptable.
So the donor came in, donated. You processed it?
No, I didn't process for that study.
But someone processed it and then they gave it to the patient that day?
I think it was later on. It wouldn't have been same day, but yes.
Yeah, a lot of logistics involved, right?
Absolutely.
Difficult.
Yeah.
So that was the first study. And then the second one was multiple sclerosis. So that's interesting. That's an autoimmune disease.
It is. And I think when people hear the two, there's not a lot of connections that come to mind. But underlying both of those conditions is leaky gut.
Leaky gut. Right. Come on. Dr. Craven, how many diseases are linked to leaky gut in your opinion? How big is this?
I think a lot of autoimmune diseases are linked to leaky gut. So including MS, things like rheumatoid arthritis, asthma, type 1 diabetes, these are all things that people have measured. There's increased intestinal permeability, which is it's interesting because they're very different diseases.
That's very interesting. That's very broad.
It is.
Oh, how does that explain? Is there some sort of different genetics involved? And if you've got leaky gut and that genetics, or those genetics, then you get...
Yeah, I think there's probably people that are a bit more resilient, that if they had leaky gut, maybe they don't go on to develop the disease. So I think it has to be a variety of factors that to line up for that to happen.
Yeah. What factors cause leaky gut? We talked about alcohol, diet, change in bacteria.
Yeah, a lot of it's diet, lifestyle. I think stress could probably have an impact on that as well.
Stress.
Yeah, are you thinking about yourself?
No, I mean, not really. Of course, it's hard running a scale-up business. It's been hard since day one. There's not been a single day where I've got to the end of the day and thought, oh, it's just so easy. My to-do list is done and I'm shutting up shop. It's just not like that. When you're a CEO, you have to provide a 24/7 service.
Yeah.
And you want your CEO, I think, to be fully engaged and to be thinking about things all the time, because there's so many problems to solve. And we've talked about things today that we've yet to solve.
Yes. Most of it,
Right?
I think.
Well,
yeah.
But the way I see it is they have to be solved. We have to figure out why leaky gut's important, how many patients it impacts, and how we solve leaky gut. We've got to figure that out. We've got to figure out dose. What's the right dose? How do we measure that? And is it the same dose for every single person with leaky gut? And if not, why? So there's all of that to think about. And it's difficult. So of course it's not a stress-free existence, but managing stress is key. And that's lifestyle and diet and trying to switch off and delegating and that kind of thing. So don't drink. If you drink a little bit. Never drink to excess, because that's not going to help your stress.
No, and it's not going to help your gut microbiome or your intestinal permeability.
Although I did read a French thing that said red wine increases diversity.
Interesting.
Yeah, of course the French would say that. Yeah. We found that cheese and red wine.
All the things that are good, yeah.
Cheese and red wine increases
diversity.
Yeah. I've actually seen that published, by the way. No, seriously.
Okay.
Yeah. So I think
I'm going to have to read up about
somebody told me at a conference that this is in France, one of the keys to reducing the hangover is to have a big, huge bit of blue cheese or very bacteria-filled cheese after you've been on a night out or drunk, because it re-establishes diversity once you've assaulted it with ethanol.
Yeah.
Because a surefire way to kill bacteria is to bathe it in ethanol.
Probably, yeah.
Isn't it?
Yes, definitely at the right concentration, yes.
They're away.
Yeah.
Yeah. I've taken us off piece again. Sorry, where were we? How big is leaky gut? That's where we were. How big is leaky gut? And you've listed a whole range of diseases.
Autoimmune diseases, more and more, I think we're coming out about that. Diseases to do with liver health, again, because that's the first organ that's encountering that blood from the portal vein. It's probably going to have the highest concentration of those inflammatory molecules as it goes through the liver, and that's the liver's job, is to try and filter those things out.
Yeah. So who decided MS?
That was, again, a very talented team of people. It would be a slightly different composition of that team, but it was a team that was looking for kind of out-of-the-box ideas for MS.
Interesting. So what happened?
So unfortunately, that study didn't get to go to completion. The principal investigator sadly passed away, we had to stop before the study was completed. So
such a shame.
It was really sad.
That's really sad.
We didn't have full follow-up for a lot of the patients, so we had a very limited data set to look at after. But in the limited data that we had, we did have some patients with MS who had improved intestinal permeability after the FMT. And that was exciting to see the result replicated again.
Absolutely. And is that the only result that's ever been published in MS, to the best of your knowledge?
I'm not familiar. I'm sure there's been more data since then. But this would have been one of the early pieces of work.
Yeah. Did you see any clinical,
safety seemed to be maintained. Yeah.
No flares? And because I guess for the listener, one of the concerns about autoimmune disease is that whilst you do an FMT and you'd maybe dampen things down and keep people in remission, so no symptoms, or put people into remission, there's also the risk, which has been demonstrated, I think, in a small number of cases, where you cause the disease to flare up, and you have the wrong, or the undesired outcome of stimulating the immune system in that way. So
the symptoms of MS were measured. There's a expanded disability status scale, which is a questionnaire that's administered to patients with MS to measure disability, and that didn't get worse throughout the course of the study.
Superb.
So safety was demonstrated there.
Superb. And I have seen more and more work on the microbiome in MS, so maybe someone at some point is gonna go for it. Now, before we move on to what happened next in your career, should we talk about probiotics?
Sure.
Because I gather that you used to work with the person who came up with the definition for probiotics. Is that
They were
correct?
part of that.
Part of the team.
That team that came up with the definition. I think updated it a few years ago as well.
Right.
Mm-hmm.
So
From Glasgow as well.
yeah.
Yeah.
He's a Glaswegian
He is.
and living in Canada.
Yeah.
Yeah.
So it's Gregor Reed is his name.
Is that right?
Yes.
Does he still sound Glaswegian?
I don't know if
Now
he'd be
that you live here.
upset. Now that I live here, there's definitely people who have a stronger Glaswegian accent than he does. Yes.
But he's still a Glasgow boy.
Yeah.
So what is a probiotic?
I need to look up the definition to get that wording exactly right. If you ever listened to it and heard me say it and it wasn't right, I'd be very concerned.
You'd say,
Yeah.
Should I try?
Sure. I don't think I could fact check you on it, but go for it.
Right. A species or strain of bacterium, it's more precise than that, but hear me out, that when administered or consumed in sufficient quantities confers a health benefit to the recipient.
Yeah.
Is that it?
I think there's some
I think-
different words in there.
Oh, is there?
I think the intention is the same.
Pretty
Yeah,
much it, right?
yeah.
So how many probiotics you can buy off the shelf meet the definition?
I think a very small proportion
Yeah.
of those do. Because
Right.
you need, randomized, double-blind clinical trial data to support that.
Yeah.
And not all of them have that.
Yeah. Very few, right?
Mm-hmm.
So maybe it's not a good idea to buy lots of probiotics.
I'm hesitant when it comes to probiotics because of that piece. There's not a lot of data that supports certain strains being beneficial. So
Do you take any at all?
I don't. Do you?
I don't. But I have.
Yeah.
I have. I
And
don't.
I have in the past
Yeah,
too.
I don't, but I have. Does
Yeah.
that make sense? Yeah.
Mhm. And I think the more that I learned about it, the more I realized how many people advertise something they say is a probiotic, but doesn't actually meet the definition. And I think it's a real disservice to the field of probiotics that so many people make these claims that
Right.
aren't true.
But there are some products that are actually supported by quite a large number of studies.
Absolutely.
So one of them is a Simprove product, which you might have heard of. And then the other one is VSL3 from Ferring. There are clinical studies supporting them. And I've not recommended them to people from a medical perspective, but they've said, look, I'm going to take a probiotic. Which one do I take?
Yeah.
And the advice is typically, well, don't mix them all together. Maybe start with one, see how you feel.
Yeah, and go with something data-driven as well.
Yeah.
If there's supporting evidence, I'm more inclined to take
Yeah,
that.
yeah. Okay. So, are we now on the part of the story where you moved from Canada to Scotland?
Sure.
Or,
Yeah,
so you got your PhD.
I did.
You
Very
did the
exciting.
defense.
I got my PhD 2 weeks after lockdown went into place in Canada for COVID. So I was one of the first defenses that was on Zoom for the university.
Wow.
And that was a challenge.
and your PhD was the 2 studies and all the work around
All that
right?
work, and then some lab work as well that was looking at the stability of FMT products in different storage conditions as well, which was really helpful. Again, I was saying, at the beginning, it was a fresh product. You had really limited constraints on how to manufacture this and give it to a patient within that time window. And the goal, we shifted to something that could be frozen, and now people shift to capsules and all sorts of improvements to the stability of those kind of products. But part of my PhD was to look at how we could improve stability too.
And did you enjoy it?
It was fascinating.
Yeah. So
I think the most fascinating bit is how resilient bacteria are.
Yeah. Mm-hmm. We've spoken loads about bacteria. We should speak about the other things, but that's part of that. So can you remind me, how did we actually connect? Was it on LinkedIn?
No, I was looking for job posts.
Did you email?
I think I emailed just general to EnteroBiotix. It was like the info at EnteroBiotix, and you were the person who replied, which was great. But I think I said, I'm finishing up my PhD soon. This company is really interesting. I think I'd be a good fit. There was nothing posted on the website in terms of jobs, but I was just like, I'll shoot my shot and see if there's anything that comes up. And you wrote back being like, oh, actually, there is a position that we're going to hire for. It was a KTP associate position between EnteroBiotix and the University of Aberdeen. You should submit your application. And I said, oh, great. And I did that.
Yep.
A few weeks later, I got an email response that said, we're not hiring for this anymore because of COVID. The project isn't moving ahead. And I was like, oh, that's really disappointing.
Yeah.
I was really excited about this. And then fast forward another few weeks, it's like, oh, actually, no, we are going to hire for this. Can you have an interview through Zoom?
Yep.
And I prepared a presentation about what my PhD was. There was a lot of technical questions I think I was asked between the interviewers that included you and different professors from the University of Aberdeen, and I thought it went really well.
Yep.
And I got a call back a week later to say, sorry, not for you. This isn't for you.
Happens.
Yep.
It happens. But
I think it was, it must have been Alan, I think, who contacted me initially. He's like, yeah, but I think James would be interested in you working for the company. I was like, all right, I'll wait to hear from him then.
Yeah. And then I was like, yeah, we'd like to find a way to make it work.
Yeah.
Because, well, the work you did was fascinating and you did a brilliant, fantastic presentation and could tell that you were very capable and driven and well, thought you were going to do great things. And then it was just a case of how do we make it happen?
Yeah.
Because we were in COVID.
Yeah. COVID in difficult times. 2 days after my interview, my dad died unexpectedly.
I remember that, yeah.
Yeah. And I reached out to you to say, I really don't know when I can make it over here.
Yeah.
But one of my last memories of him is him prepping me for that interview and
Really?
how excited he was for me.
Oh,
and
wow.
when I got to tell him how well it went, he was just so proud.
Wow, wow. What a nice memory.
Yeah.
So we made it work. We managed to get you a consultancy arrangement and we worked on all sorts of things like building a database of FMT studies, right? And that was quite fun. And then it was, how do we actually get you over here to work, right?
Yes. You were sending me all sorts of different visa options.
Yeah.
And
the fastest one
was the global talent visa.
Yeah,
and I remember you sending me this and I was like, I really don't know if I'm good enough for
I
this.
don't know if I'm global talent. I was like, yes,
I know.
you are.
It's a really big title to say
yes,
you're globally
you are.
talented. I remember I looked it up and I think the first one that came up was like the arts and humanities category. It's like, you should apply for this if you have an Oscar. Or like something like this. I was like, I don't think I'm an Oscar winning equivalent scientist. I don't know if this is for me.
That is so funny.
Yeah.
But one thing you know about me is that if I want something to happen and I believe in it,
it manifests.
It manifests.
It does.
It manifests.
It does.
I was just like, gotta make this happen.
And you did. I needed a letter from you, I think.
Yeah, and a letter from a few other people.
Yeah, a few other people. People I have never met to this
With
day.
big titles, right?
That you had to write some letters for me to support me. And then I included all my publications in the assessment too.
I won't say who it is, but I actually managed to get, there's only there's a very small number of people in the UK who are like Grand Order of the Knight Cross or something like that. They're not just a Sir, they're a Sir plus. They're like,
wow.
They've been like honored to the absolute highest level. And I got one of those people.
That is what I needed.
I got one of those people. And they said, to whom it may concern, did I fully support Dr. Craven? Did it? And you know what?
It worked.
It worked.
It did.
And we got you the global talent visa.
Forever grateful for that.
You're welcome.
It's worked out very well.
You're welcome,
but yeah,
you're welcome. Yeah.
No, that took a bit of time too.
You have to, sometimes you just got to go for it.
You do. I
just got to go for it.
Just sent an email, looked at the website, thought this would be a really great place to work, and then it all just worked out.
The universe runs its course and what have you. So you came over and then you're in this new country and I remember, I think your partner was somewhere else, was in New Zealand or something.
He was.
And then he was coming over.
Yeah, it was a long year. I know. So he was in New Zealand for a year because the borders shut down. And my plan after my PhD was to go travel New Zealand for a few months,
right.
And that was going to be great. But then obviously COVID happened, couldn't go anywhere. He couldn't get into Canada, so it's like, what countries could we be in? The UK, and everything just kind of lined up. He had flown over to move here, and I think the Alpha variant happened at the same time, and then all the flights in and out of the UK stopped for 3 weeks. So he was in the air between Hong Kong and London when I found out my flights were all cancelled and I wasn't coming.
And you were in Canada?
Yeah.
No.
So he had to land in the UK, and I had to make the call. I was like, I'm so sorry, but I can't get there. I've made you move halfway across the world. New Zealand, the safe haven from COVID.
Yeah,
and I've
no
sent him to
lockdown.
the UK.
No
and
lockdown.
I know.
Wow.
And it was another 3 weeks before I could make it here, but it all worked out.
It all worked out. And now you're still together. And
Yes, getting married in 5 weeks.
Whoa.
I know. So another thing to thank EnteroBiotix for as well is that we got to be together and it's all worked out. So I feel like there's been a lot
Not
of
only are
great
we
experiences.
facilitating global talent visas,
Yes.
but also facilitating marriages. You get married in Scotland?
Yes.
How wonderful.
Yeah.
That is fantastic. Is he going to wear a kilt?
I don't think so, only because he's not Scottish. I don't
you
know.
should get him to wear a kilt.
Yeah.
You should. You should get him to wear a kilt.
Yeah.
Oh, that's so nice. Fantastic. Good. I'm really happy for you. So what kind of stuff are you doing now then at EnteroBiotix?
So I've recently been promoted to lead research and development scientist, which is very exciting. So I do a lot of study, experiment design, analysis of data, trying to make improvements to different processes and analytical techniques, providing advice for clinical aspects, IP, business development, that kind of thing. So I feel like
Busy.
I get to be involved in a lot of aspects of the business and it's really exciting.
Good.
Yeah.
What's exciting you the most at the moment about all the things you're doing?
First patient dosed is
Yeah.
very, very soon. I think that's what I'm most excited about.
Yeah, 100%.
Yeah.
Getting the trial underway and yeah, I'm most excited to see the data,
Yeah.
because we've been working on this now for 5 years and I really, truly believe we have a best-in-class product. I actually think we have the best full-spectrum product in the world. And I think we've got the most scalable manufacturing process. And it's all in-house. So that, for me, is a recipe for success.
And things people told you couldn't be done.
Yeah,
And
of course.
you've done it.
A lot of that.
Yeah.
Yeah, well, that's just standard. Yeah.
Yeah.
That's just standard when you're trying to do something that other people haven't done. It's very easy to critique and say why something's not going to work. But I guess it's my job to be like, actually, nah, we're going to figure it out. We're going to make this happen. We're going to do it. We're going to overcome whatever obstacle comes in our way. And now we've got such a good team. We've got such an awesome team of people who are just so smart, who've actually overcome a lot together.
Yeah.
If you think about the journey the company's been on,
Resilience. We're very strong in that. Yeah.
over the last couple of years, there's been so many ups and downs, and that's just part and parcel of the roller coaster that is biotech and drug development and being a small to medium-sized enterprise. But everything that we've faced that's been difficult, we have overcome. We've found a way to we've figured it out, and even when it's been like scratching our heads, what are we going to do? Or you see those things in the films where like the hay.
Yeah, the tumbleweed. Was past. Yeah.
We're like, what are we going to do? But we figure it out.
Yeah.
So that's awesome. So tell me about your own experience with the microbiome. And I know you've got a YouTube channel where you talk about your health.
Yes.
So tell me everything. Well, not everything. Tell me whatever you want to tell me.
Sure. So after I started my PhD, I think I was aware of kind of being a bit hypocritical that I'm here talking about the gut health, but I probably didn't have the best diet in the world. And I became vegetarian a couple years into my PhD. And that was driven by a lot of reasons, but one of them was the microbiome and gut health.
Okay. And is vegetarian the best diet?
I don't know. It's the best diet for me at this point in time. And that was really my focus. I don't know if it's the best for everyone, but that's what worked for me.
So when someone asks you, what do I need to do to improve my microbiome? What do you say?
Eat more fruits and vegetables, eat less processed food. If there's anything I go by in life, it's eat real food, mostly plants and vegetables, and not too much. And I feel like that covers most aspects of healthy eating.
Yeah, it does. And it's almost like healthy eating advice is the same as healthy microbiome advice. Yeah. Isn't it?
I think it goes hand in hand.
It's the same thing. So on your YouTube channel, you talk about your journey with type 1 diabetes.
Yes.
You feel comfortable talking about it?
Absolutely. Yeah. So I was diagnosed with type 1 diabetes 4 years ago. So this was after I'd gone vegetarian. And it started after having an Epstein-Barr virus infection, so glandular fever. So I had that, and then about a month later started having the symptoms of diabetes. And being the busy PhD student that I was,
running to the loo a lot, waking up at night.
Yeah, I think I was needed to pee like 3-4 times throughout the night, drinking liters and liters of water, couldn't maintain weight, felt like I was eating constantly and it made no difference whatsoever. And so I felt really unwell. But I don't know, because I felt like I was so busy trying to do my work, I just never took the time to look after myself. So it took a few months for me to actually go to the doctor and get that diagnosis. And even when they told me that, I was like, oh, surely no. Like this, it couldn't be me. So
when you explain the symptoms to the doctor, so I'm getting up all the time,
really thirsty all the time, frequent urination, low energy, weight loss that can't be explained. I think they knew immediately what it was. They knew. And we're trying to protect my feelings. It was like, we'll run a panel of tests, we'll get back to you. So I had my blood work done.
What did you think at the time? You just thought you were busy, stressed.
I had the logical mind to know the symptoms meant diabetes. But I think being diagnosed with a chronic illness is a lot like going through the stages of grief. And the first stage is denial. And I kept thinking to myself, this can't happen to me.
It's not me.
Yeah.
Fascinating. Fascinating.
So he called me back within 24 hours of my blood work. And it was, your blood sugar is very high. Your A1C is very high. I've made you an appointment with the endocrinologist that's in the building. He'll see you in a couple of days. And I was like, okay. But he never said I had diabetes.
And then you went
And in my mind, I was like, nah, it's still not diabetes. But obviously was. So I went and got that diagnosis, started on insulin that day, had a glucose monitor that day.
Wow.
My blood sugar was 30.
How was that?
Intense.
It was 30, so the normal range, I'm going to get this wrong.
It's 4 to 7 usually.
I was going to say that. I was going to say it's 4.7, 7.4. No, is that potassium? Oh my God.
I don't know about potassium,
formal
but
medical school formal medical school colleagues are looking at me like this guy doesn't
4 and 7, and mine was much higher than that.
What was yours, 30?
30. And I felt very unwell.
That was high.
Yeah.
That was high. So you're in the room and the person says you've got type 1 diabetes.
It's a state of shock.
How did it feel? Yeah, it's a state of shock.
I think I was really trying hard to process that information. I think it's really easy to take your health for granted. I was 25 at the time and I kept thinking to myself, I think I have this healthy lifestyle and diet. I felt like nothing could ever impact me, which was wrong. This was something completely outside of my control, something I couldn't prevent.
And there's this person saying, look, you've got to take this insulin shot now, and you're on it now.
For the rest of time.
For the rest of your life, unless we find a cure.
But at that point, it's like, this is something you're going to have for the rest of your life, and that's a heavy weight to put on someone.
It is a really heavy weight to put on someone. So how did you manage it, and how have you been managing it since then?
I manage it like a scientist, and I think I went into my coping mechanism, which is like data collection mode, which is I want to read a lot of papers, I want to read a lot of books, I want to talk to experts, I want to talk to people who have this to try, what can I do to like hack this? Can I be the best person ever to manage diabetes, which is a really unrealistic goal to put on yourself, but really helpful for managing illness. So I just tried to do the best that I could. So you've got to take a certain amount of insulin for the number of carbs that you eat. And I was experimenting with different ratios and just trying to...
I'm really sorry, really sorry to cut you off. But can we just go back a step and you can explain to the listener what type 1 diabetes is? Yeah.
So type 1 diabetes is an autoimmune disease. And the beta islet cells in the pancreas get targeted by the immune system. And one of their functions is to make insulin. And your body can't take up carbohydrates or glucose without insulin to aid that. So when you have type 1 diabetes, you're not making insulin anymore because these cells have been destroyed. And glucose builds up in the blood and just kind of spills over into urine if you're not being treated appropriately with insulin. And it's something that damages a lot of cells in the body and has really poor effects if it's not treated.
And I guess it's like basic science, but why is it that having too much glucose in the blood is a bad thing? Like what happens? It blocks like small blood vessels. So anything that has really intricate blood supply, like your eyes and nerves and your kidneys, things that have those small blood vessels, those are the first to be impacted, and it can affect a really wide variety of organs in the body and be quite damaging. So it's really important to get that glucose to the normal range for your...
And that's the goal, is to spend as much time in the normal range as possible.
Okay. And talk us through your journey of sort of self-exploration.
Yeah.
And what it means on a day-to-day basis in terms of planning your food, planning your work, planning your insulin.
Yeah, so I eat the exact same thing for breakfast every day since I've been diagnosed because I want one meal that I never have to wonder what's going to happen to my blood sugar after. And it's nice to have that consistency. And it's yogurt and granola and berries. And I always have this for breakfast.
Somebody told me that you make your own yogurt. Is this correct?
I have. I haven't done it since I've lived in Scotland, but I have made my own yogurt in the past, and it's a really fun activity.
Do you make your own kombucha?
I've never tried.
Do you know who's trying? What is it today? Friday. Do you know who's trying tomorrow?
No.
Me.
Oh, you doing this at home?
I'm going to kombucha masterclass.
Oh, that's great.
Yeah. Yeah. This Glaswegian company makes kombucha. And they're doing a masterclass tomorrow for 2 hours.
Is this You and I?
Yeah.
The company You and I? Yeah.
Yeah. Not You and I.
Not You and I. The company called You and I.
It is.
Yes.
It is. They sent an email like 2 months ago being like kombucha master. They do good kombucha.
They have excellent kombucha.
It's so good, isn't it? It's actually so good. And I know we've stopped, but I won't say that. We've stopped stocking it in the office for a while as part of our wonderful finance director's
it's a responsible decision, but I miss the kombucha. Yeah.
It was the premium product.
It is really nice.
It was so good. I just loved it so much. What was your favorite one, just out of interest?
The lavender one.
The lavender.
That was really nice.
Yeah, I do the lime and sea salt or the lavender.
Yeah.
But rest assured, when I go tomorrow, I'm absolutely going to be saying, please, can you give us some free kombucha?
Well, now you can make kombucha for the office.
Well, yeah. Well, we'll see. Let's see. I'm actually in a really nerdy way. I'm really excited to see like what is their like facility like.
Yeah.
Because we've built facilities, and I just get really excited about how do you make kombucha? How do you scale kombucha? Sorry,
that's great. But in terms of
you've not made your own yogurt since coming to Scotland. Is that because we're not able to do it in Scotland, or just
no?
Too busy?
I need to buy the equipment to do it.
Got it.
Yeah.
Okay.
But
good wedding present for anyone listening.
I love that. Oh, that's great.
Shout out anyone listening. Lindsay Howard.
Oh, it's excellent. It was one of my lockdown hobbies. I didn't have a lab to be in, so the coping mechanism was to make my own yogurt.
Yeah, okay.
Yeah.
Okay.
And it was fabulous. Loved it.
Yogurt, fruit, granola, every day, same amount.
Yep.
Perfect.
Easy to predict, same amount of insulin, start off every day, consistent, no kind of concerns about how the blood sugar is going to go. And that's what I like to do to prepare for work, I know exactly what to expect. Work isn't going to be interrupted. And I like that a lot.
That is important. Mm-hmm. And just for argument's sake, or just hypothesize, or if you don't have that, how do you know what the effect of something's going to be on your blood glucose?
It's really difficult to predict. I could eat the exact same thing, every day for a week and have a different blood glucose after, even though it's the identical meal, because there's so many other factors that can impact your blood glucose.
Like stress?
Stress. Yes.
That great old thing.
I know.
Stress.
I had my blood glucose monitor on for my PhD defense, and I could watch it just continue to increase the whole time. And then as soon as it was done, come right back down again.
Because the cortisol?
Yeah.
Oh, that is fascinating. Mm-hmm. That is so interesting. Wow. Okay, so stress is a big one.
Stress is a big one.
Yep.
In making sure you have enough sleep, being hydrated, things like that. So I think one of the best things that came out of being diagnosed with diabetes is that I really brought taking care of myself to be a primary focus of my life.
That's really good.
Mm-hmm.
So stress management, more consistent exercise probably than I did before, being really mindful with what I eat, eating less processed food, less carbohydrate heavy food that didn't have a lot of nutritional value associated with it as well. So I wouldn't want to have type 1 diabetes. If I had the option not to have it, I would take that. But there's been a lot that I've gained from it as well. And a lot of that is having a healthier lifestyle, I think. That's a really positive outlook to have on it. And it sounds like you've taken away a lot of great things that you might otherwise wouldn't have.
But
it's quite inspirational.
For me, it's trying to find foods that are foods I enjoy eating, first and foremost.
What are they?
Falafel. I do love falafel, yeah.
I love falafel too.
It's really good.
Falafel and salad bowl.
That's
just
nice.
Have it every day.
Yeah, but also being mindful of the number of grams of carbs in it, trying to balance with different proteins and fats to feel full. Because as a vegetarian, I think it's really easy to have a carbohydrate-heavy diet. But when I got diagnosed with diabetes, I kind of had to adjust to have more protein, more fat. And a lot of it's just trying to keep track of those things too. There's a lot of my day that's spent reading labels, writing things down, doing the math to calculate the grams of carbs, doing the math to calculate how much insulin I need to take. So,
and other apps and tools that can help you with that kind of thing?
Yeah, there are.
Yeah.
I've got an insulin pump as well, so I can use my insulin pump. I just program in, I'm going to have this number of carbs.
And it gives you...
And it just gives me a number. This is what they suggest.
So the insulin pump, it goes subcutaneously all the time?
Yes.
Just like sits in like... Where does it sit? Like
you can move it about. So I have an Omnipod, and the infusion sets last for 3 days, and after 3 days you throw it away and you put on a new one. So mine's on my arm right now, and it's the big device that's here. Yeah, you can't really notice it.
Notice it.
No, they're very subtle.
You would never notice it, and as you said. So it sits under the skin in the adipose tissue or like the fat cells.
Yep.
And there's a small needle that goes in there.
There is. It's a cannula and it's plastic, so it's actually flexible. So it doesn't really feel like there's anything stuck under the skin.
Does it hurt or?
It can. Like the biggest issue I have is when I sleep on my side and you can feel that pressure on it. But I don't usually feel it at all.
So you must have an insulin requirement at night as well, right?
It's all through the day. So there's one requirement is when you're eating, and then there's one that's just little amounts all throughout the day as well to keep it balanced.
So there's different types of insulin.
There
fast acting, longer acting.
Yes. Yeah. When it's in a pump, it's all fast acting, and it just gives teeny little amounts every hour.
Oh, interesting.
Yeah.
Interesting. So what are your hopes for diabetes? Like, do you keep up to date with the developments and things like that?
Yeah, I think just now what would make the biggest impact is accessible and affordable diabetes care.
For everybody.
For everyone around the world. And I'm really grateful to be here with the NHS because all my prescriptions are covered, the insulin pump is covered. But when I was in Canada, these were things that cost me a few $100s a month to purchase for myself.
Wow.
And that's a really big burden to put on someone.
Wow. So someone's not working.
Even if you are working as a student, I couldn't afford all those things. I needed help.
Oh, wow. So in Canada and presumably the US, you can't, they don't cover that, or like the healthcare system? The healthcare
system, so I'll just speak about Ontario because it differs for province to province in Canada.
Right.
But when we say universal healthcare in Ontario, that's just really doctor's appointments and specialist visits. It has nothing to do with prescriptions.
Wow.
So you would be responsible to pay for your prescriptions.
Wow.
Now I don't know if it's most people, but many people would have a private health plan, and it would cover either the total cost of your prescription or some proportion of it. So as a student, I had a student health plan. It paid for 80%, and I needed to pay for the other 20% out of pocket. And
goodness gracious,
20% of some of these things is a lot of money.
That's a lot.
Yeah.
So that's important. We spoke about a cure. There's also a lot of people trying to develop oral insulin as well, I believe.
Yeah, different formulations.
Yeah. So, what are your hopes? Do you think there could be a cure? And if so, what are the most likely avenues, do you think?
I think a lot of people are talking about some kind of transplanted cell, like having islet cells in some kind of like implanted device, or that could just make your own insulin for you. Wow. I think that would be very exciting. Other people are trying to regenerate the cells that people have lost in their body. Okay. There's been interesting FMT data, which I know we want to get back to.
We've got to get back to that, yeah.
And I want to talk about that. But there's a period in type 1 diabetes called the honeymoon phase. So when I was diagnosed, I probably still had quite a few cells in my body that could make insulin still, but it wasn't enough to cover my needs. And that is the honeymoon phase, is when you're still making a little bit of insulin yourself, but it's not enough. And I required very little injected insulin to manage my blood sugar at that time.
Understood.
But there's people doing, I was about to say FMT, doing IMT studies that are looking to prolong that honeymoon phase. So what can you do to preserve those cells that you still have left to make insulin?
Yeah.
Because it's a lot easier to manage diabetes when you're making some of your own, and it's better health outcomes that way too. Yeah. So can you prolong the honeymoon phase? And if so, how? And
the vision there is honeymoon for the rest of your life, if you can.
If you can, it would be great to revert to having enough insulin so it's not diabetes at all. But if not, the honeymoon phase is still better. Yeah.
So a lot of people talk about genetics and type 1 diabetes, but my understanding is that actually there's a stronger link to type 2 diabetes genetics, Or maybe I've got that wrong.
I think there is. I think there's still a genetic component to type 1. So you've got a higher risk if one of your parents or your siblings has type 1. But I think having family history of autoimmune disease just would put anyone at risk of type 1 diabetes as well.
That makes sense. So should we talk about the FMT studies then?
Yeah, please.
Well, over to you. IMT, sorry.
IMT studies. There's people who have
really
given IMT, and it shows that you can prolong that honeymoon phase, which is really exciting. So I'm sure there's a few trains of thoughts for why it's working. My theory is that you're giving an IMT, it's having a big effect on the immune system, and the immune system is too busy dealing with the IMT product to be thinking about the pancreas, and maybe it leaves it alone.
There was one very interesting finding in that study. Well, there's a lot, but one of the interesting findings was that the autologous group, so the group that got their own, there was some bigger impact.
Yeah.
It was why could that be? Do we know?
I don't know. I think
because I spoke to one of the UK's leading experts in this, and he asked me.
Yeah, he wants you to know.
He was like, "So why is that?" And I was like, "Sorry." I'm asking you the question. I was like, I'm asking you the questions. You're the expert.
Yeah.
But, well, I guess for the listener, this expert was actually very excited about this study on the basis that FMT is safe and IMT can be administered alongside other medical therapies.
Yes.
And might have like a durable, long-lasting effect because you're just instating, you're allowing these factories to just stay there and produce what they're producing, and that somehow has a dampening effect on the immune system. But that was a really interesting finding, which goes against your findings.
Yeah,
I know it's different, but
it's different manifestations. So the patients in the study I was a part of would have had metabolic syndrome. So they were still able to make their own insulin, but it wasn't in sufficient quantities because they had insulin resistance, some of which would have had prediabetes or type 2 diabetes.
This makes sense. Is there leaky gut, you believe, in type 1 diabetes?
I think there is, and it's been documented as well. So they have measured intestinal permeability in patients who have type 1 and patients that are healthy, and there is elevated intestinal permeability in type 1, which is interesting.
That's very interesting. Epstein-Barr virus?
That's what I had.
What's the link between EBV and type
EBV
1?
is linked to a lot of conditions.
Is it?
Yeah. But a lot of people report getting type 1 diabetes after having an infection. And there's something about that really robust immune response that seems to have triggered diabetes after. And there's quite a few people who've been diagnosed with diabetes after COVID as well. And those publications are coming out now that there's a higher incidence of type 1 diabetes potentially due to COVID.
Well, prior to this podcast starting today, we were talking, with the recording starting, we're talking about strep throat and psoriasis. Because it is a very classical picture where someone has a streptococcal throat infection or some sort of upper respiratory tract infection. And then they get a specific type of psoriasis called the guttate psoriasis, which looks like little like raindrops all over their body. Usually resolves within 3 to 4 weeks. 3 to 4 months. And my thinking is that, so what's actually going on there? You know, I think that the upper respiratory tract bug somehow finds its way systemically.
And it's easy for that to travel to the gut just from that route. That's where it would naturally drain as well.
It would go, so it would go through the body and it would find its way through, somehow bypasses the defenses, and then maybe through some sort of leaky gut,
bacterial translocation,
it finds its way and it sort of causes the immune system to go wild across the body. Who knows? But leaky gut, I think. Leaky gut, could be the thing.
It could be.
You do eat dairy because you eat yogurt, but you know there's a lot of talk around dairy irritating the gut. Is that something you believe in, or do you think it's a bit of like, it's not a real thing?
I'm not going to say it's not a real thing. I think there's definitely people that impacts. I don't know if everyone's equal in that. I don't know if it would have the same impact across the population, or if there's more people that are potentially more susceptible to that.
Because some people with autoimmune disease say that it makes things better.
Yeah, and if that what's
they
worked
cut
for
dairy.
them, then go for it.
Which
Yeah.
is a shame, because obviously halloumi and other forms of cheese
Yes.
and yogurt are all dairy, and it's tough. I've tried it for a while. It's tough. It's really tough. In one of the early episodes, there was a nutritionist and gut health influencer that came on, Christian Servidis, and her gut reset program involves cutting out dairy, cutting out gluten, cutting out coffee, cutting out alcohol, and I'm hoping that's it. Yeah, for like...
Was there no sugar as well? Was that something?
No, I don't recall.
Okay.
I don't recall that. I think definitely gluten, definitely dairy, definitely alcohol, coffee. Did I say coffee? I said coffee.
I think so.
Coffee, I mean...
Yeah. That's tough. I don't drink coffee, so for me that's pretty easy, yeah.
Have you never drank coffee?
No.
Wow. Ever?
I've tried it, but...
You don't like it?
No.
What about tea?
I don't really drink tea either.
Oh, man. Your morning routine is a good one. Mine does vary a little bit, but one thing that's stayed true is coffee.
Yeah.
Not as soon as you get up though, because you need that adenosine to flush out. You don't want to be blocking that, and sort of artificially waking yourself up. But it's good. And Debbie Shawcross said that the more coffee you had, the better for your liver. That honestly, I promise you, it's protective for lots of. Apparently, it's protective for lots of different things. But if you don't like the taste, then you don't like the taste. I think I do like the taste.
I would hope so, given how much you drink.
I don't drink it that much.
Is it less? Wow.
I know what you're thinking now. I know what you're thinking. You're thinking back to those days when we were in the same office.
I just watched you make coffee all day long, I think so.
Things have changed.
I'm proud.
I've dropped it down to one.
Wow.
One a day. Not 3 or 4.
Yeah.
Or 5. I think that was the max. I think I never got above that. But I know a lot of people that have 3 to 6 cups a day. But I'm just on 1 now and it's all good. And I look forward to it because it's just nice to have 1 coffee a day. Anyways, so looking into the future, where is the microbiome field going in your opinion?
We're getting closer and closer to a mechanism of action, I think. And there's been lots of...
For FMT.
For IMT.
Oh my goodness, can you please?
For IMT.
Please.
And there's lots of conditions that there's been beneficial effects, found. But there's still no real clear reason why. Yeah. And I think looking ahead, we're going to find out that reason why and be able to have more targeted products that aren't the full spectrum anymore, like an IMT is, and a more targeted approach to achieve that mechanism of action.
So let's have the debate. What if it's not the bacteria? That means all those defined consortia companies, they're finished. Or hold on a second, or it's not just the bacteria. For reasons that we don't fully understand. Yeah. For it to work durably, you actually need some phage in there, and you need some fungi in there, and you need some
metabolites of some kind.
So and you need some archaea.
I think the bacteria are one of the easiest things to measure, and that's been the main focus of this kind of data. Yeah. But you're absolutely right. They're not in isolation. They're making all sorts of different material. There's lots of other microorganisms that are present, and more and more people are researching and looking at that data, which I think is exciting. So
why are you so confident that we're going to replace IMT with the bacterial fraction and not the viral fraction or the fungal fraction, the archaea? No, seriously, why is this? I'm not trying to grill you. I'm just like, seriously.
It's genuinely, that's what I have the most experience in. So when I think about it, I'm always thinking about the bacteria. I mean, it's my own bias as a microbiologist, as a bacteriologist, that I'm interested in the bacteria. But you're absolutely right. There's so many other components at play. And I'm sure we're going to get to a day where we think, oh, why were we only thinking about the bacteria? There were so many other things that were important here.
Right. And I suppose, just to support your case, they are metabolically active.
They are.
Some, the virus element isn't, clearly.
It can still have metabolic effects, but like a virus in isolation, not metabolically active.
Yeah. Very interesting. what if it's the case that it's not possible to continue to take such a reductionist approach? I mean, why do we need to take a reductionist approach? Do you believe that a defined, refined, reduced diversity product could be more effective than the full spectrum?
If it was targeted, maybe.
Targeted to the mechanism of action?
Yes. So right now, because we give everything that those few components, or maybe many components, that's what's necessary, are in lower abundance than they could be if the whole product was just made of that.
I understand that.
Yeah.
And therefore you get less in the way of mechanism of action. Okay.
Yeah.
But that assumes that the mechanism of action is singular or is simple enough for a consortia to act on all the important components.
And that's what we don't know. Yeah. Because there's so many different connections, and I've seen those schematics where someone's made all the connections between everything in the microbiome, and you just think, how on earth can we replicate that in the handful of bacteria that could go into a consortia?
Can I also just push back a little bit and state that the microbiome in all of our patients, or not our patients, but patients, is different?
Yes.
Therefore, you're likely to encounter different resistance. Absolutely.
I think there'll be certain microbiomes that are probably more easily treated with the product than others.
Got it.
Because there's a lot of variation even within a single disease. There's a lot of variation just in healthy people about the gut microbiome composition too.
Yeah. I mean, I am biased because I'm a CEO of a company doing full spectrum, but but I think full spectrums.
I think it's the right place to start.
Yeah, I see. I believe it's the gold standard. I do. Or a variation of the full spectrum is the gold standard. It's like we're using the body to treat the body. I just think that there's just so much that we don't understand in the microbiome. And it could be the interaction between the microbiome and the host. It could be the most complicated thing in the universe. It actually could be. In the context of human health.
Yes.
That's because of its complexity, it really could be. So we should be accepting of that fact, perhaps, if it is a fact, and trying to figure out how to replicate the studies that have been conducted, but make them better. And I believe that the best way to do that is through IMT. But I also hear what you're saying. If there's a mechanism and it's defined and it's clear and it has a significant effect on the target, then you might want to go more refined. So how do you go more refined? What's the approach? If you're in charge of it?
Trying to identify, maybe through engraftment, what bacteria or other organisms have stuck around. So I know you might have a different idea of what engraftment is, but I'm going to say mine, and I'd be really welcome to hear yours after. But I think of engraftment as two different types of engraftment. Primary engraftment are bacteria or other organisms that are in the material that was delivered, so a product, FMT, IMT, that are in the patient after the fact. And then secondary engraftment are organisms that weren't in the patient before, aren't from the product, but have now found a niche in this new environment that have appeared. And what about you?
Sounds really good. Yeah.
I wasn't expecting that.
Why were you laughing before? Because
I said engraftment, and I was expecting some kind of pushback that you had a different idea of what it was, because you just started laughing. And no, I know you've had discussions about engraftment with other people, yes. But I mean, the reason I started laughing is because I know that you know that I like having debates about engraftment. Yes.
And it's just, I find it, that's why I was laughing, because I think you knew as soon as you said engraftment that I was likely to be triggered.
Yes. No, absolutely. So
oh gosh.
I think part of it is looking at engraftment data and seeing what is there.
Yeah.
Could we just deliver that component? Is that what's being helpful? But it doesn't take into consideration, transient effects from taking the product, things that are in such low abundance that maybe we can't detect, and all sorts of the metabolomic profile and the other impacts that it's having. So I think that's a very simplistic approach would be to look at engraftment.
Yeah, I'm with you. And I would assume then that the observed effects need to be pooled across the whole study, and you need to see if that engraftment,
And
quote unquote, happens in the same way across every single person.
Yeah.
And then you've really got like a smoking gun, right?
I think so. That would be really clean data if that's how it came out. I think more often than not, you're going to see very different things that engraft, but potentially their functional output is the same. And
exactly.
Exactly.
The functional output is the same because of the functional redundancy.
Yes, I love that. I love that bit.
Lots of different bacteria can do the same thing.
Yes.
Which is why my microbiome is different to your microbiome, but it's doing a lot of the same things.
Absolutely.
And vice versa across every single person on this planet.
Yeah.
And I believe that's another tick in the box of the full spectrum. You got more shots on goal.
I agree with that.
Yeah. So engraftment is one way.
Metabolic output, those pathways, I think is another way to look at it.
Is it fair to say that even if the microorganisms that are engrafting is different, at an ecological level, there's consistency? Like, for example, increasing diversity.
Yeah. And again, at the functional level, two very different bacteria, could have basically the same output. You don't have to have the same thing. So I think it'd be interesting to look at that data, and maybe it doesn't look the same across different people, but when you look at the function of the microbiome, has it all kind of come together and aligned?
And in your expert opinion, is increasing diversity ever not a good thing?
Depends on the body site. There are certain sites of the body that you would not want increased diversity.
That makes sense.
Yeah. But in terms of the gut, with the data that's available, increased diversity seems to be protective of health in, acute cases of recovering from disease, but also in terms of longevity, where people who are making it past 100 years old have the diversity of someone in their 20s who's healthy. And that's really interesting.
That is fascinating. And that could be a result of their diet, or it could be that they're literally living to that age because of something we don't understand, which is that the microbiome just stays true.
Yeah, and I think what's missing, that missing piece is longitudinal data. Are they living that long because of the microbiome? Is the microbiome like that because they've made good, healthy lifestyle, diet decisions? And you really can't get that without a very large longitudinal study.
And you're touching on one of the key issues in the microbiome field, which is what comes first, or what's driving what.
Yeah.
Is the disease or the disease processes driving the changes to the microbiome, which we see? Or is it the microbiome changes that are driving the disease processes and making people sick? Or is it vice versa? Is it
or somewhere in between. I don't know if it's, exactly. So
the chicken and the egg conundrum,
Yes, trying to find people before any disease is manifested and tracking them throughout life to say, what is driving disease? Is it the microbiome?
Yeah, exactly. So when people come to you and they ask you about FMT and they say, what's FMT going to be used for next?
I'll start by correcting them that it's IMT.
Yeah. Good. Oh my goodness, yeah. I've actually done an episode on IMT and I'm still calling it FMT, but it's just entrenched in the brain.
It is, and I think it's entrenched in public perception as well, and that's a negative consequence on the field.
You're right.
It's putting people off something that has the ability to save lives.
Oh, absolutely. Yeah, absolutely. I totally agree with you. So, sorry, IMT. What's the next indication or the patient population?
Next indication, so many have already been explored, but they've all been such small studies. I think what's next is having larger phase 3 studies to try and get more than a safety signal. I think a lot of them have started off with, is this safe and tolerable in patients? They've proven that it is. Can you now see a clinical benefit after that?
Yeah, I agree with you. I think we need well-powered big studies with placebo control. But the problem is that's typically done by the pharmaceutical industry, not by the academic community. And then we get to this crossroads of, well, you can kind of only do that when you've got a drug.
Yeah.
Whereas the classical academic studies involve just IMT that's been prepared by people in the lab. I guess not too dissimilar from yourself when you were doing your studies. And I think that work's really valuable though.
There's so much value in that, and I think it's really helped industry focus on what the most likely indication should be, and that's from all sorts of academic work that have taken those big risks and looked at all different kinds of indications to provide us with that kind of data.
100%. I totally agree with you. So it's like the academic groups generate the proof of principle, the proof of concept, show there's a signal, just like in that diabetes study, and then the industry can look at that and go, okay, that's promising. Maybe we can develop a standardized composition and take that forward. Because I think one of the other challenges when you're comparing all these studies is that, we talk about standardization, that there isn't really truly standardization, is there?
No.
Because there's different protocols and the product's slightly different. And
it is. And even if you're relying on human stool, even within a single person, their composition of the microbiome can fluctuate over time.
How much does it fluctuate though?
I think it depends on people.
Yeah.
I wish I could have measured my microbiome throughout life and just watched it change. But I've had big events like becoming a vegetarian, going through that state of hyperglycemia before my diabetes diagnosis, following diabetes diagnosis, moving to Scotland. I don't think I have that different of a diet, but there's different food that I just can't buy here. Different like fruits and vegetables that just aren't available. Oh, like what? Oh.
Is this a bugbear?
Oh no!
Was there not some crisp that you couldn't buy that was a problem for a while or something?
No, there's no.
Was it not? Was that a joke?
Probably.
Was that a joke?
There's a lot of different fruits and vegetables I think are more common in North America than they are here. There's like a lot of different squash varieties that I really miss that I can't buy here, things like that. So I think that has an impact that I'm not getting the same types of dietary fiber. I swear every few months here there's a shortage of fruits and vegetables for whatever reason too, I think.
No!
I just never see those some days. There was a few months ago. I swear there was.
Yeah, there was one. Yeah. I know.
It has happened.
Yeah, there was one. But that was a global thing, was it not? Or was it just the UK?
It feels like it was worse here for whatever reason.
Like it was worse, yeah. No,
things like that happen. But I think I probably have slightly different eating habits from being here. But not that they're any detrimental to my health. It's just a slightly different variety of food.
Glasgow's got a reputation for some very interesting types of food.
Yes. I haven't tried the pizza crunch yet.
All of it's, deep fried stuff.
Deep fried.
It's not, you know me, it's not for me. It's just not for me. I don't like it. I'd like more falafel and Buddha bowls, but we don't have that much here actually.
I think even restaurant options. I'm used to being able to order a main salad at a restaurant full of all sorts of yummy stuff, lots of protein, whatever else. And you go here and the salad is a side salad and the vegetarian option is a plate of 100%. Chips. Yeah.
It's not good. It's not good. That honestly is one of the biggest bugbears I've got about Glasgow and Scotland. You've just hit the nail on the head. It's like the healthy options are not readily available.
It depends on when you look. There's definitely restaurants where I would have an easier time looking, getting what I'm looking for. I think.
Sure. And we should compare notes after.
Yeah.
Because it's Friday and I might go for dinner.
I miss going to a restaurant in North America and I know they're going to have a giant salad that's full of lots of delicious things, lots of protein and very filling, wide variety of things on the plate.
Colourful.
Yes. And knowing that is readily available and no one's going to give me a funny look when I ask, can I have a big salad?
I sympathise. I know what you're talking about. Mm-hmm. Sometimes you just need a big salad in life. Yeah. Good. Do you have any other scientific interests outside of microbiome? Is there anything you keep tabs on?
Main focus other than microbiome is type 1 diabetes.
Aliens. No.
It's not the aliens.
It's not aliens. No.
You promised there'd be no aliens. But it's type 1 diabetes and the different advances in the field. And a lot of it's just like engineering different technology and what goes into that and the kind of electronics and tech side. I think that's fascinating what people can do to create an organ essentially that you can control yourself to take the place of the pancreas.
Where does the organ go? Did they just put it back to where the pancreas is? Or what's the thinking there? It just sits somewhere in the body and produces insulin?
Yeah, I think that's part of it. But for me, the insulin pump is taking over some of the roles of the pancreas, and that's like its own little organ as well.
And it's getting better and better. Than
it is, and they get more efficient. They are using AI to predict.
AI.
I know, that's really cool.
So they're predicting when you need and when, what you need and when? Or?
So you can get insulin pumps that will connect to your glucose monitor and adjust your insulin for you. You don't even have to do anything yourself, which I think is, that to me is the future.
So
if there is a
cool.
day where I don't have to think about this and it can just take care of itself, that is the dream to me.
So you just scan all your food and, the resulting thing is you don't have to input anything. It just scans and it knows. Or some are even more sophisticated. You just eat it and it knows.
That's just it. I would want to just eat it and it would just figure out what I need.
It measures your glucose and it responds instantaneously.
Yeah.
Have you looked much into this AI explosion that's happening right now, the chat GPT and things like that?
It's really interesting what it can write responses to, especially concerning like scientific data, what it's able to generate. I think that's really cool.
Are you using it in your work right now?
No,
Sometimes, no?
I don't use it in my work.
Some people are.
Yeah.
Seriously, like top academics are using chat GPT or GPT-4, whichever the latest one is. I think it's GPT-4, to write manuscripts.
Wow.
To help write. Yeah. It's really,
that would make it so much easier.
It does. It really does.
Yeah.
And in some respects, I think it's great because it's increasing productivity. But in other respects, it's like, wow, that's quite scary. Like, I've asked it hard questions. Is it listening to me now?
Probably.
It is. It probably is listening.
I'll have ads for it after on my phone because it knows we've been talking about it.
Almost certainly.
Yeah.
One day it will scrape this and listen to it. So I just want it to know that I'm its friend. I've asked it very hard questions about IMT.
Oh,
yeah.
Interesting. What did you
and
ask?
Microbiome. I asked, what's the next indication for IMT?
And what did it say?
I asked why companies have failed clinical trials. I've asked it to summarize all the available evidence for IMT in ulcerative colitis. I've asked it to summarize all available evidence for IMT in Y and Z. Some of the answers have been really good. Really good. Like to the point where I'm, you know how it like sort of comes up in front of you and it just sort of like populates and you're like, wow. That's like, it's almost like an expert's typing it as it's coming through. But then some of the other answers were just very bad. I told it. I said to the thing, that's a very bad answer because of this. So it's probably learned now.
It's still learning. Interesting.
But it's scary. I'm scared of it. I am, honestly. Truly am. I think it's immensely powerful. And we're just letting it happen. We're letting it happen. And it's consuming so much information. I mean, I listened to a podcast with the CEO of OpenAI, which is the company that runs GPT-4 and ChatGPT. And I'm pretty sure they asked the question of him, is it conscious? And he said, I don't know. So it might be.
Yeah.
That is just
that's scary.
freaky stuff.
I know.
Freaky stuff. Because I know we all get the ads on our phones, but that's a totally different thing.
That's a different level.
That's a different level. Anyways, I know we've gone way off piece. Sorry. AI
in diabetes tech is very exciting though.
Okay, we've been on for an hour and 40.
Sure.
We've talked about a lot. And I need to record your intro and things like that. So probably call it.
That sounds good.
Dr. Laura Craven, it's been a pleasure.
Thank you very much for having me.
Thank you. Thank you very much. Very interesting conversation, and hope the marriage, wedding day goes very well.
I'm sure
I hope you get your yogurt making kit from whoever's listening to this as a wedding present.
Thank you for dropping the hints. I'll be keeping an eye on my deliveries to see if something turns up at the door.
And thank you for all your hard work and excellent work at EnteroBiotix.
Thank you. Thank you for the journey of coming to EnteroBiotix, and now you know what a big role it's played in my life.
I do.
Yep.
And it's humbling, and thank you very much.
Thank you.
Cheers.